You ever wondered how Dolly the sheep was made? In this video lesson you will learn about the process of cloning DNA. When you hear the word clone, you may think of the word sci-fi movie or perhaps something more realistic like Dolly the sheep. But DNA cloning goes far beyond this. DNA cloning is taking a piece of DNA from the organism where it's naturally occur and putting it into a cloning host.
Well, let's take a look at the steps involved in the cloning process. First, the scientists interested in cloning will isolate two kinds of DNA. One is the gene that has the DNA that will be cloned, that is your DNA of interest or can be called as insert, and the other is a bacterial plasmid that will act as the vector of the DNA cloning. Insert can be prepared by a few methods which are PCR, restriction enzyme digestion, DNA sonication, fractionation by agarose gel electrophoresis, and chemically synthesized oligonucleotide.
A vector is a small DNA vehicle or an agent. Your vector should have compatible ends with the insert, be able to carry the foreign DNA and act as a replicon. The most commonly used vectors are plasmid, phage, and cosmid. There are more types of vectors such as bacterial artificial chromosome, yeast artificial chromosome, and retroviral, which have their own pros and cons.
After isolation, the scientists will treat both the plasmid and the gene of interest with an enzyme that cuts the DNA called a restriction enzyme. These enzymes get their name from their role in the nature to restrict invading DNA from entering bacterial cells by cutting out the foreign DNA. This invading DNA may come from other organisms or even viruses, so it's important to keep them out. There are three types of restriction enzyme: type 1, type 2, and type 3. Type 1 restriction enzyme, it cuts both the strands at a non-specific location, approximately more than 1000 base pairs away from the recognition site. The most common restriction enzyme is type 2, where it cuts both the strands at a specific, usually palindromic, and recognition site at about 4 to 8 base pairs. Whereas type 3 cleavages at one strand only and 24 to 26 base pairs downstream. The restriction enzyme cuts the plasmid in one place so that it creates an area that the target DNA can bind to. The target gene is cut out of its original DNA strand so that just the gene of the interest is attached to the plasmid for cloning. After cutting both the target DNA and the plasmid, the two are linked together with an enzyme called DNA ligase. This pasting process results in a recombinant DNA plasmid, which is a single DNA molecule combined from two different sources of DNA. It is literally recombined, hence the name recombinant.
After the two DNA pieces have been pasted together, the plasmid is inserted into a bacterial cell or a host in the process called transformation, which then will allow the bacteria to replicate and produce plasmid babies that are identical to the parent plasmid. For transformation, the first parameter is the method used to induce competence. Competent cells are capable of uptaking DNA from their environment and expressing DNA as functional proteins. Calcium chloride treatment is one of the best methods for the preparation of competent cells. The second measure is the genetic constitution of the host strain of the organism being transformed. Basically, the hosts are usually bacteria because of several reasons. Well, in this video, we stated as 5 main reasons. The first one is E. coli is a single-celled organism. There are no articles concerned about growing, manipulating, and killing bacterial cells, unlike multicellular model organisms like mice or chimps. They are also tiny cells. So, in a small laboratory, you can have plants containing billions of cells that take up very little room, allowing many experiments. Secondly, they reproduce and grow very rapidly, doubling every 20 minutes. So in 10 hours, you will probably get 30 generations and approximately 1,000 million descendants. With bacteria, it is as easy and fast as letting them grow overnight. Trying to study the same process in the subsequent generation of elephant, for example, will require several generations of scientists and more elephants than we have on the planet.
Next, they can survive in variable growth conditions. As we discussed earlier, this leads to it being very adaptive yet forgiving in lab situations. Culture media containing simple and inexpensive ingredients and nutrients can successfully spur E. coli to grow and thrive. Most naturally occurring strains of E. coli are harmless, as food poisoning E. coli that sprang to mind earlier are the exception, not the rule. When scientists first started using E. coli for lab experiments, they chose a strain that was harmless, means that studying E. coli poses a little threat to researchers and the public. Lastly, bacteria can be genetically manipulated very easily. E. coli especially is very well understood and can be readily manipulated or engineered. Thus, our clones are born.
Created using Powtoon.